Welcome, everyone, and thank you for joining us today. This is PPMD's webinar with Sarepta. We're very happy to have Doug Ingram and Teji Singh, their Chief Medical Officer, with us today to discuss what their future looks like and what plans we can see going forward. It's certainly been challenging times in 2020, and we hope you are all staying safe and healthy, and we're looking forward to hearing from Doug and Teji about their next steps. So with that, Doug, I'm going to turn it over to you.
Thank you very much, Pat, and thank you all for this opportunity to respond to some of your questions and provide some update on where we are with our gene therapy for Duchenne muscular dystrophy. And that gene therapy, just to remind us, is called SRP-9001. I am also very pleased to have with me today Dr. Teji Singh, who is at Sarepta, is our Executive Medical Director of Gene Therapy. As if you stump me, Pat, I can always turn to Teji, who can't be stumped with respect to this therapy. Next slide. I'm not sure, Pat. I don't see the next slide popping up yet.
There you go.
There we go. Yeah, I will. I won't read this. You'll all be very thrilled to know. But it is a reminder of the fact that I'm going to be making statements today that may lean into making some kind of predictions about the future or what I expect may happen in the future. And it's important for us all to remember that, I can't predict the future, and that the statements I made when I make them are my belief right now, but of course things can change dramatically, and there are a lot of risks with that. So we should all speak cautiously about the future, including the future of our therapy, and be guided by the evidence and the data that we have in front of us today. Next slide. Let me start by explaining, I'm sure many of you already know this, but for those who don't, I will explain in very broad strokes what gene therapy is and what it's made up of. And I'm really focusing right now broadly on gene therapy for Duchenne muscular dystrophy and microdystrophin, but with a specific focus on our construct for gene therapy. So just to remind you all, gene therapy in this regard doesn't intend to integrate with your genome, with the patient's genome, or to edit the gene. So, of course, as we all know, children who have Duchenne muscular dystrophy have one of a number of potential mutations on the gene that codes for this structural or shock-absorbing protein dystrophin, and as a result of that, they're not protected when they move their muscles. We don't intend to go in through this gene therapy and correct that mutation. We go in and we ignore that mutation. That's what this kind of gene therapy does. We deliver, if we're successful, into the muscles throughout the body, and that's the skeletal muscle, diaphragm, the cardiac muscle, a separate gene that sits next to the genome, doesn't integrate, but starts making a form of dystrophin to replace the dystrophin that isn't being made by the mutated dystrophin gene itself. And there are three components. The first is the delivery device, or what we call the vector. And what we use here is something called RH74, and we're alone in using RH74. Others use a different form of vector. And let me explain what a vector is. It's essentially a re-engineered virus. And so for everyone who's been dealing with this horrible pandemic over the last 8 or 9 months, we've all unfortunately been getting lessons in how viruses work. Viruses are good at two things, basically. They're good at getting into cells. They've through natural selection in eons, they figured out how to get into cells, and then they hijack the machinery of the body to essentially make more of themselves. And they get into the cells through something called a capsid. It's essentially a ball. You've seen, frankly, pictures of this coronavirus ball that makes up the coronavirus capsid. The good news is we don't use a live virus for this. What we've done, what science has figured out over many decades now, is to re-engineer a virus, to take out the virus, that part of it that can make more of itself, the thing that would actually make you sick, but leave behind a shell or capsid. And in this case, for us, we use something called RH74 that can act as a delivery device to bring this therapy all over the body, hopefully, with these full body infusions. And we choose, and I say we, really, this is led by brilliant people Dr. Jerry Mendel at Nationwide Children's Hospital and his partner there, who is now our head of gene therapy, Dr. Luis Rodino-Clayback, chose RH74 for a host of reasons. One is it's very tropic. What does tropic mean? It means that it's very good at getting into heart and skeletal and diaphragm muscle. It also, at least in animal models, looked like it was associated with very good safety profile, which we can't overstate how important that is, of course. And the final thing is that at least what we've seen so far and what we saw in animal models in advance is it is correlated with a very low level of what's called pre-existing immunity, which means if you have, along your journey in life, interacted with something in the environment that would look like this vector, in our case, RH74, then you could have pre-existing neutralizing antibodies that would unfortunately mean that you'd be screened out from getting this therapy, at least as the science is right this minute. We can talk about that later. But the good news, at least so far with RH74, is it has so far demonstrated a very low level of pre-existing neutralizing antibodies in the community and in patients. So we're talking something in the mid-teen range that potentially can get screened out, but the great majority of people would not have that issue. So that's the way we chose this vector or delivery device. So going to the left, there's this thing called the transgene. The transgene is the gene that starts making the protein that we make. In this case, it's microdystrophin. And I'm going to talk on the next slide about we're making microdystrophin and we're not making dystrophin. And then finally and ours is, I will say, ours is unique to our program. Every program has a different approach to these genes. I think Luis has done a lot of really fabulous design work and experimentation to come up with the gene that she's choosing. And then finally on the right, we have something very clever called the promoter. The promoter is something that attaches to the gene, and it essentially tells the gene when and where to turn on. So oftentimes, a lot of this viral vector or capsid will go into the liver. The promoter will tell the gene, don't turn on here, you're not in the right place. But when it gets to a skeletal muscle or diaphragm muscle or cardiac muscle, then the promoter says, you're here now, you're at the right place, start making the protein. And in the case of our promoter, which is unique to us, we think we've made a very good choice with a very robust form of this promoter. Next slide. And so before we get to this, let me explain what we do with gene therapy. These are full body infusions. We send, essentially the therapy throughout the body in the hope of getting this capsid to bring this therapy and deliver the gene to muscle all over the body. We'll talk later about the challenges associated with that and what we've seen thus far. But let me first answer the question that may be in some people's minds, which is, why are you using some form of dystrophin? Why don't you just use the full-length dystrophin that would normally occur? And the reason for that is that the gene, I'm sure many of you already know this, the gene for the protein dystrophin is the longest gene in the body. It is enormously long. The capsid, these so-called AAV capsids, are not very big. And so they can only carry so much material into the body. And the gene for dystrophin would be too big to fit into this capsid. In fact, it's about 3 times too big. We would need a gene that's about a third that size to comfortably fit into this vector called RH74 and to be delivered to muscle. This is how it all came to be. On the far left of the slides in front of us, we'll see it. This is a muscle with a full-length dystrophin protein attaching to it and attaching to what's called the sarcolemma or the muscle membrane and essentially acting like a shock absorber in the muscle. Something fascinating occurred in the late 80s, and in fact, I think it was 1990 was the actual publication, where a 61-year-old gentleman who was still walking went into a doctor's office because he was having some issues and was wondering what was going on with him. He got genotyped, and lo and behold, he had what would essentially be a Becker muscular dystrophy. He had an enormous mutation on his dystrophin gene, but the gene itself was still made a messenger RNA that was in frame, so he made dystrophin. The dystrophin he made was a fraction of the size of full-length dystrophin. It was about half the size of full-length dystrophin, and this was a gentleman who was still walking in his 60s as of the time of this publication. And that got people thinking, and among a lot of other people that got thinking on this was Dr. Luis Rodino-Clayback and Jerry Mendel as well. And they started some work some long time ago of design work and then testing it empirically in animal models to see if they could come up with a gene that would code for a protein that would remain functional like the full-length dystrophin protein, but could fit inside an AAV capsid. And you can see this on the far right. That is our protein. That is a graphical representation of the protein that our gene makes. And it will not surprise you to see how similar it is to the middle image. And the reason that it's so similar to the middle image, of course, is that Luis and team started with this case study as the basis for this, because of course that 61-year-old had a greatly truncated form of dystrophin, and yet it was clearly functional. And that's where we came up with this. And I'm doing a poor job of describing in detail how much work went into this, both design work, design features, but also empirical testing to come to a place where Luis was confident we had a dystrophin that could be both functional and safe if it was delivered to muscles. Next slide. So we did a proof of concept study, and we started this proof of concept study a couple of years ago, actually. I think the first boy, if I'm not mistaken, was dosed in 2018. And I'm going to talk about this study, and I'm going to talk about these results, but I really want to make sure that I'm careful to point out that this is only for patients, and that whatever we see here has to be confirmed in a well-controlled clinical trial. And in fact, as you'll hear, we're doing that right now. We're in the midst of that trial, and I'll talk about that. So as is normally my way, I will tend to get enthusiastic, but I really want everyone to understand that what we're seeing here is a proof of concept, and we need to really prove out what we're seeing in this well-controlled trial, and we're in the middle of a blinded trial now. But let's talk a bit about what we did. We dosed in an open-label 4-patient study, 4 patients, and they were 4 to 7-year-olds. And there are 5 things that we have to look at to determine whether the therapy appears to be working in these kids. And of course, as you can see on this slide, the very first thing we need to figure out is that it's safe and tolerated. Because if it's not safe, we're out of the game. We have to go back to the drawing board. The good news, at least in these 4 kids, is it was safe and well tolerated. You can see the data here on the slide. I will say 3 of the children had elevated liver enzymes. That's not uncommon with respect to these full body infusions, because the liver does capture a lot of this capsid. But also the good news is that all of this was transitory. The children's, all 3 of these children's elevated liver enzymes very rapidly resolved back to baseline with just an increase in steroid treatment. So broadly speaking, the therapy was very, very safe and well tolerated, at least in these first 4 children. Let's go to the next bullet. The next bullet was, from my perspective, probably one of the most significant moments for us, because when we started this journey, what we're trying to do is no small feat. This is not like an injection in the eye of a gene therapy. We're trying to do a full body infusion to get therapy, to get this gene that we've made, this gene construct, to muscle all over the body, all the skeletal muscle and the diaphragm and the cardiac muscle. So that was a big question. Can we actually get it there? Will this capsid really deliver the gene robustly like we hoped and like what we frankly saw in animal models? And the good news in these first 4 children was that it did. In fact, we were delivering not simply robust, but very robust 3.3 genome copies. Essentially, for every muscle fiber on average, we were seeing about over 3 of these genes in every muscle. Really strong indication that we were getting a good gene to the right place. The next question, of course, is, okay, that's great. Can you make the protein that we're trying to make? And if we do the next click, we'll see the answer to that. And the answer to that was yes. We were making, at least in these first 4 children, on average, an extraordinary amount of this form of dystrophin, this microdystrophin. As you can see here, we were making what is approaching normal amounts of dystrophin. At least this is microdystrophin, but normal amounts. We were between 74% unadjusted and 95% if you adjust for fat and fibrotic tissue using Western blot, which is the way we quantify dystrophin. So we were really excited about that. The next question that we asked ourselves is, okay, is it getting to the right place? That's an enormous piece of information. And we used something called immunohistochemistry to try to figure that out. Here's what you see on these slides are the immunohistochemistry results in biopsies for these first four boys. On the far left, that biopsy is from a non-Duchenne muscular dystrophy age-matched child. And all of the red that you see there, that's dystrophin located at the muscle membrane, acting like a shock absorber. So we wanted to know, are we seeing dystrophin, and is it at the right place? Is it at the muscle membrane? And what do you see on the right? The top part of the slide is a biopsy pre-gene therapy, and as it won't surprise you, you see nothing on the slide. Those are basically without any dystrophin. On the bottom of these slides across these children, you will see this enormous amount of dystrophin lighting up in immunohistochemistry, and as you can see, all properly located at the muscle membrane, meaning that at the place where it can act like a shock absorber and protect these children's muscles when they move. As you can see, we had about 81% on dystrophin-positive fibers, meaning on average, about 80% of the fibers had dystrophin up on them. And then what's called intensity, which is 100% is what that child on the normal control would have. We were seeing intensity levels of dystrophin in these biopsies of about 96%, almost the intensity of a non-Duchenne muscular dystrophy child, at least in these first four children. So then we looked at function, of course. Now, this is where we really have to be careful because this is four children. It is open label. It's not blinded. And so it's very easy to fool oneself. But we will at least say that we were very excited about what we have seen across these children. I won't go into a ton of detail on these slides other than to say this. There is a JAMA neurology publication that came out in June of this year that reported the 1-year data for these four children. And in this 4-patient open label study, all children were outperforming both the baseline and the natural history on every functional measure, NSAA, which is the primary measure, but also the times tests across all of the children and the composites. Then we went back and we were able to look at the 2-year data. These children have now been on this therapy for 2 years, and I'm excited to say that they continued not only to maintain what they had already gained in the way of milestones and functions over that period of time, but they actually had some modest gains even from 1 year to 2 years. So we were excited to see that as well. Next slide. So as I said many times, and I'll say yet again, we have to be led by the science. Yes, I'm excited about what this might mean, but we need to understand that we have to prove this all out in well-controlled trials. So I want to be careful to be very scientific about that. And so we've done that, and we are in the process of doing just that. So that first, on the first far left, you'll see that's so-called trial 1. That's the first 4 patients. The very next thing we did was we have to start a trial with a larger cohort of children, and it has to be blinded, and it has to be placebo-controlled. It has to be powered to show whether this therapy is functional or not. So we don't just get hope out of it, but we get data that we can use, hopefully for the purposes of working with regulators to bring this therapy to children around the world. That trial is right now, it's called Trial 102. That's a 41-patient trial. It's in 2 centers right now, one in Nationwide Children's Hospital with Dr. Jerry Mendel, and also out at UCLA as well is a second site for this trial. That is a placebo trial. It's 4 to 7 years old. For those who may wonder, why do we keep talking about 4 to 7-year-olds? To get this therapy to all children, we need to make sure that we can prove to the satisfaction of ourselves, and then equally important, of course, to ministries of health and regulators around the world, that the therapy actually is benefiting children, is transformative like we hope. And so we have to narrow the age range that we look at to make sure that we can see a difference in a relatively short period of time. And so we narrow that down. Now, from a mechanism of action perspective, of course, this dystrophy should work across all ages, but that's the reason we're looking at the 4 to 7-year-olds, and we'll talk about what we're going to do beyond that for other children as we track towards approval, because we're obviously not making a therapy merely for 4 to 7-year-olds. So that's study 102. The great news about study 102 is that all of the children for the main analysis have been dosed. Their functional results are being taken. It's blinded right now, so I don't have information to provide on that, and I wouldn't take anything away from, I wouldn't read into any of my enthusiasm what that study might do when it reads out, because we won't know until the study reads out. But it will read out, and we will publicly disclose it early next year, and that will be an important moment for all of us, and I think, frankly, for the Duchenne muscular dystrophy community, because we will have in the well-controlled trial both evidence, if we're successful, the efficacy and the meaning of this therapy, as well as additional evidence on the safety. Now let's talk about the studies on the right of this slide. In the summer of this year, we made some choices and decisions over the course of this year to keep this program moving, even in the face of this COVID pandemic, which has certainly been a challenge on all of us, but has been a real challenge to these patients in clinical trials right now. This summer, we had been contemplating about a 75-patient study, something we called Embark, or Trial 301. And that study was going to do a lot of things at the same time. And we're going to do all of these studies. But that study at the time was intended to be another confirmation of a placebo-controlled trial. It was going to be multi-centered. It's going to be around the world, in Europe, in the U.S., in Asia, as an example. But there's going to be a particular feature about these next studies, the ones in purple, that are different than the blue studies. The blue studies, 101 and 102, have been done with what's called clinical material. It's been material that was made for us by Nationwide Children's Hospital in their GMP facility. But that process that they make that material with can be scaled to serve the community commercially. It doesn't scale so that we can bring it to all patients. It's fit for purpose for these smaller studies, and that is obviously our goal. So we have spent an enormous amount of time over the last couple of years investing in and building capacity and a new process to make this material in a way that's fit for purpose to launch and to fully serve the community in the United States and Europe and around the rest of the world as well. And we've entered a partnership with an extraordinary company, Roche, which is a large international company that can help us bring this therapy to patients even beyond the United States and to the rest of the world, and do so rapidly. But what we need to do is dose children with our commercial material so that we can show what our underlying data would suggest as well, which is the commercial material acts the same way as the clinical material. So this summer we were looking at what we were doing, and we realized we needed to divide these trials up so that we could get Study 301 going at the right time in the face of this pandemic. But we needed to start a separate, smaller trial to make sure that as early as possible next year, we could get data with the commercial material in hand that showed the performance and validation of that material, both from an expression perspective and from a safety perspective. So this summer we proposed to the agency, as we were tracking into the fall, that we wanted to do the Study 301. We also wanted to do the Study 103, which starts at about 10 patients. And we also want to do something called Study 303. Now, we're going to be doing a number of different studies on older and non-ambulatory patients as well. Trial 303 would be a large multinational trial for non-ambulatory patients, and we're looking to do the same kind of thing with non-ambulatory patients that we're doing with Trial 103 as well, which is an open-label study to show that the material provides the same kind of expression and safety, and do that in a very rapid way with non-ambulatory and older children, just as we're doing that right now with this current age group. The issue is when we went to the agency in September to get their blessing, and I won't go into a lot of sort of the technical details, the agency disagreed with our perspective on the particular release assay that we were using. It sounds technical, but it's important. And they disagreed with the particular release assay that we were using, and therefore we couldn't start any of our studies. And there was a real risk associated with that, I understand, because if the agency is very busy right now, the FDA in particular, this pandemic has only made things more difficult, but already the division that deals with gene therapy and what's called cell therapy, which is related, is probably approaching about 2,000 different drug INDs that they're looking at the same time. And our big fear was if we didn't get approval from them rapidly, that it might take months or even longer than months to eventually come to an understanding with them that would allow us to start a trial using the commercial material. And we've got to use the commercial material as any predicate to a discussion with anyone about getting this therapy approved so we can bring it to a much larger group of patients and hopefully, if it's successful, benefit the Duchenne community at large and not simply small pockets of the Duchenne community through clinical trials. So I'm going to give the team immense kudos. The team moved heaven and earth. They were able to convince the agency to have an informal meeting about 2 weeks after the formal meeting that we had where we were not permitted to continue. And we went into that meeting with a very specific goal. We wanted to find out what the agency's view was on our release assay. We wanted to solve it with them in the meeting, and we wanted to start this study 103. It was important for us that we had that win out of this meeting because we need to dose kids on this commercial material, and we need as early as possible next year to have data that shows that the commercial material, as we will predict, acts the same way that the clinical material would predict. It is essentially the same underlying construct. It's just made in a slightly different way than the clinical material. I'm proud of the team. We went into this meeting with that goal. It is very difficult to get meetings like this with the FDA. And I also want to give kudos to the FDA for their willingness to be flexible and have this meeting. And we went into that meeting and we did come out with the goals that we had asked for. We found out what the agency's view was on release assays. We proposed an alternative approach, which they accepted. And then we convinced them that we should be able to start this commercial material trial called Trial 103 as soon as possible. And we will start that this year, before the end of this year, certainly. And so what does that mean? So we're doing that right now. We will still start this larger Trial 301. That trial will start after we get an initial read on Study 103, so it won't be delayed very significantly. One of the concerns we had with a large multinational trial that we were considering in the summer was that we're in the midst of a pandemic, and if there's a second wave, it could really put people at risk. We have that issue, of course, unfortunately, coming to fruition with the second wave. But our goal is to start Study 301 as soon as we reasonably can, and we can talk about that. We're also going to start as soon as we can our non-ambulatory trials, and we have a number of them that we're looking at right now. We want to start a trial very similar to 103 on non-ambulatory patients. We need to talk to the agency and get their blessing on that. And we want to start this larger study, Trial 303, on non-ambulatory patients. That's going to be a global trial as well. And again, we need to work on that and get the agency's blessing to start that trial as well. But those are all very front of mind for us. Because as I've said, and I think there's always been consternation around this, the goal of all of these trials is to have a broad label that allows access to children with Duchenne muscular dystrophy without regard to mutation and without regard to age. And that is certainly our goal, and that will continue to be our goal, and we're tracking in that direction. Next slide. This is broad strokes, the timeline. We will start this Study 103 for commercial material this year. So, it'll be in the next month. We'll be dosing children with Study 103. In the first quarter of next year, of 2021, we will have the results for Trial 102, and we will be releasing those results. Those are extraordinarily important. As I've said before, our original proof of concept study on these first 4 children really not only excited us about the potential for SRP-9001, but also placed upon us an enormous sense of obligation to move as fast as is possible to confirm those results in a well-controlled clinical trial, and we'll have those results early next year. I'm excited about that. We're going to plan our open-label non-ambulatory study, and we're going to start that as soon as we can get that planned and we can get the division to concur in that. We'll start that as soon as possible. And then, of course, we'll start study 301 next year. We'll be able to announce the results of study 103 in the second quarter of next year. And we'll be starting next year as well our non-ambulatory large or multi-center trial, multi-country trial for non-ambulatory and older children as well. Next slide. These are, I will leave this to you, but these are some places where you can learn more about our microdystrophin program and get some additional educational brochures if I have been less than lucid in what I've been saying here today. Next slide. And then, of course, here's some places where you can get questions answered about any of our approved therapies. We have a number of approved therapies, or about any of our clinical trials, both for SRP-9001 or any of our other trials. And I would write this down, which is our advocacy email for our advocacy group run by Siobhan and Allison, who can answer your questions and provide you guidance to the extent that you need information about us or any other program or clinical trial or frankly a currently available trial. The one thing I will say, and you haven't heard me talk about it, is, well, okay, that's all great, but what's your pathway to getting approved, both in the United States and around the world? And the short answer that I'm going to tell you is that I can't give you a certain pathway yet, because I alone don't make that decision. First, the science has to cooperate. Good news is, so far it is, but we need to see the results of the next trial. And then, of course, we need to sit down with those who regulate us, the FDA and then the ministries of health around the world, who are indeed the watchdog for public safety, to talk to them about the fastest pathway to bring our therapy to children with Duchenne muscular dystrophy who are waiting for it. But at least know this about us. We understand that time is not on the hands of the patients and their families who are currently living with Duchenne muscular dystrophy. And frustrating though it may be at times, but with respect to pace, we wake up every day with the idea that we've got to find a way to keep our pace as fast as the ferocity of the disease that we're trying to fight with not only SRP-9001, with our other therapies. So at least know, while I can't tell you with precision what exactly the outcome will be of our discussions with the agency, it will be our goal next year to sit down with the agency if we have positive results from our therapies, and if we've been able to show that our therapy is effective and functional and benefiting children and slowing the progression of the disease or arresting it, and that it is safe and well tolerated, and that our commercial material acts the same way our clinical material. Know, please, that we will sit down with the agency first in the FDA and then around the world and talk about the pathway forward, with the idea that we should have the fastest but appropriately fast pace to get this therapy to families that are waiting for it. And with that, I know there are a lot of questions. So I think maybe, Pat, we ought to make this a dialogue now as opposed to a monologue.
Yes, thank you, Doug. That was a very nice introduction of the topic for today. And there are so many questions. So I think I'd like to go back to slide 7, if you will. We go back there, and that's slide number 7. Okay, great, thanks. Excellent. So this was the beginning, Doug. We were so excited to be part of this initial study when you began, and so as we all entered the space of gene therapy, just sort of a little bit anxious about what this would be and what kind of things we would see. And so one of the things you mentioned on this slide is that some of the kids had experienced vomiting after. Is that consistent across what you're seeing in studies? Was it worrisome? Did it lead to anything else? Was it easily addressed in the study itself?
Vomiting was not a concern. I mean, obviously you don't want it, but it has not been a concern. It's not a serious adverse event. It's been very mild. So, that isn't going to be a significant issue. Even with respect to the elevated liver enzyme, I would say the one adverse event you see is these elevated liver enzymes. But as I've said before, with increases in steroid use, they came back down to baseline very rapidly. And in fact, we changed the steroid protocol for what we call Study 102, our larger blinded trial. And there is some possibility that we even avoid the elevated liver enzymes or much of the elevated liver enzymes simply by a change in the steroid protocol. So again, you don't want to take 4 patients and then assume that you can make firm conclusions about safety. But I would say we were very, very pleased with the way that this therapy was tolerated. And I will also say the families that participated with these first 4 children were really heroes that we all, they were real pioneers and heroes. And thankfully, I think it's broken the right way for them. But it's helped inform Study 102. We're pretty excited about what we might see out of 102.
Sure. And I know you adjusted your steroid protocol during this study. So what is the steroid protocol that you have put in place? Can you describe it, and will it be throughout the following on studies?
Yeah, so I think that it will likely be the same for all of the studies going forward. As a predicate to being in the trial, you have to be on steady steroids for 12 weeks. We are very flexible about that. So, for instance, I know some patients are on daily steroids, some patients are on weekly steroids. We don't dictate around those issues so that we're accommodating the current standard practice, and we don't force people down a certain pathway. And then immediately before the infusion, the steroids are modestly increased. I think, if I'm not mistaken, and Ted, you're going to always stand by to correct me if I'm wrong. We're from about 0.5 milligrams to about 1 milligram for post-infusion, and then they maintain on that steroid regimen for 60 days, and then they taper back down to the 0.5 milligrams per kilogram.
So do they taper back down to their original dosing structure, or do they taper from the 1 mg per kg back to a 0.5 mg per kg? So what is the sequence? Ted, maybe you want to comment on that?
Sure. So I think, Pat, they taper back down to their original pre-existing steroids. So most kids that are on daily are about 0.75, and if they're on a weekend, they're on a slightly higher amount. But after that 60 days and the 30-day taper, they go back to what they started before the infusion, back to their normal regimen.
Yeah. Do you ever see a day when there are no steroids following a gene therapy?
Yeah, and I think that's the goal. I mean, we don't know yet. We would never, you've got to be careful not to confound things. But of course, it depends on what we see with this therapy over time. And I don't think even at the launch of this therapy, that's where we'll yet be as a community, as a people. But, one could envision the day where a gene therapy was sufficiently profound that you could avoid the impact that long-term steroids have on these children and their families.
Great, thank you. So can we go back to slide 10, please? There we go. So this expression is pretty incredible, and I think it warms our hearts. Having a dystrophin on board is sort of the holy grail. Can you give us a sense of when these biopsies were taken in this first study?
Yeah, the biopsies were taken at 3 months, and that is the standard protocol. We've found through a lot of great work from Dr. Regina Clayback and her team that 3 months is the right time to look for dystrophin.
Yeah, and what muscle was this?
This one, Teji, you can help me on this one.
This was the gastrocnemius muscle, so the lower leg, the gastroc.
Great, thank you. And in this study, this was the study in which the parameters of the mutations were children that had mutations within exon 18 to 58. Is that right?
Yes, that's right.
That's great. So could we go to slide 11?
I want to be clear about that, so we're clear about the limitations. So there was, out of abundance of some early caution, there was an elimination of some of these rare exons that occurred on either side, mutations that occur on either side of the dystrophin. gene. Upon further analysis and review, that really was an unnecessarily cautious approach. And we don't have that limitation on a go-forward basis. Now we do. I'm going to just sort of make sure we're on the same page, as you'll have seen it in a slide. When we're looking at therapies to test function, you will see that there are two mutations that are excluded from the clinical trial. They will not be excluded from the label. That is not the goal at all. But from the clinical trial, we exclude exon 44 amenable patients with an exon 45 deletion, and then these exon 8 children with, I can't remember, I think it's a 2 through 7 deletion, because, typically those children have a different path than other children, and the risk is that if, you create a trial and you confound the results, you could have a grand therapy, but you've created confusing results that don't show, like, for instance, a statistically significant difference, and you've only put at risk the ability to get the therapy to as many children as possible. So we have that in situations where we're looking specifically at function. We have those limitations, but that limitation, please don't read that as an intent to leave those children behind. We had absolutely no intention of leaving those children behind in the approval of the therapy.
Yeah, thanks for that explanation. Can we go to slide 11? So in here, this is pretty incredible, a change in the North Star. As those of you on the phone will probably already know, the North Star is a global assessment of an ambulatory child using 17 different activities that they perform. When I believe it was Dr. Montoni who, during the World Muscle Society meeting, talked about the fact that children, as they grow, usually gain 2 points on the North Star per year, and then they plateau and lose 2 points. So your mean change of +7 is pretty significant, and we're thrilled about that. I think one of our questions in this community has been, these are very young boys in these studies, the 4 to 7s, often referred to as the littles, right? And not the babies, then the littles. As these children are certainly staying stable for 2 years, I think one of our concerns is, if you see this as these boys grow, do you foresee a time when they'll need to be redosed? And what kind of mitigation strategies do you have around the idea or the need for redosing should that occur?
Yeah, that's a really important question. So for those who may not know this, and I maybe should have mentioned it, one of the things that gene therapy is today a one-time therapy. And there's two reasons why it's one time. One of the reasons is hopefully it is only needed once, that it's an enduring therapy that provides a benefit over the long life of a patient who has a gene therapy. But there's another reason. And the other reason is because currently you can only give it once. And the reason for that, let's be very clear, the reason for that is that we all have what's called a secondary immune system, which is if we've never seen some particular foreign body before, our body will slowly wake up to recognizing that it's an issue, and then we'll build neutralizing antibodies to that, and we'll be ready for it next time. That, for instance, to give everybody sort of a metaphor example, that's what we're going to be doing with vaccines for this COVID virus, right? The goal here is you give someone something that teaches the body to create antibodies that will attack this COVID virus if it ever came back again. If you give someone a gene therapy, the body initially, if it has never had an exposure before, it won't wake up immediately, which means by the time it's waking up, the capsid gets to the right place and it inserts the gene, and the gene can start making the protein, and then eventually the body wakes up, it makes neutralizing antibodies, but by that time it's fine because the gene is in place and then the capsid is chewed up and everything's great. However, if once you've done that once, the second time you would come back for that therapy, the body's ready for the therapy. And the body would attack the therapy as soon as it got into the blood and the plasma, which means that you would not get additional expression. And in fact, you would probably just create safety issues without a concomitant benefit. With that said, there's a couple things to understand about gene therapy. Generally, that's what I explained, but also about Duchenne muscular dystrophy. The one good news about Duchenne muscular dystrophy and gene therapy is this. I mentioned earlier how difficult gene therapy for Duchenne is, because you've got to do full body infusion to get it everywhere. The one potentially good thing is that once there, as we know, muscle doesn't turn over significantly unless it's been the subject of assault and the like. And so we should see long-term durability. Now, we don't have long-term durability in patients. So this is probably one of those ones where you may look back on that forward-looking statement and recall that we should be limited by the data in front of us. We have very good animal data. In animals, we see very good long-term durability. So we have lots of reason to believe that this therapy, if it's functional and beneficial and we get high expression, will be very durable. But that's something we'll see over a long period of time. In animal models, we've been able to look out to 8 and 9 years, and we don't see a diminution in durability across the various animal models that we see. So we're very excited about that. Additionally, though, Pat, we're working on this issue, and we're not alone. So this is one of the limitations of gene therapy is the inability to redose, and another limitation of gene therapy is that some percentage of children can be screened out because of pre-existing neutralizing antibodies. A lot of smart scientists are working, even as we speak, on the concept of finding a way to redose those patients. And in addition to lots of other people looking at this, we have a number of different programs in the organization right now. They're still in the research phase, so they're only hope right now. But we have some very interesting approaches that, if they're successful, could potentially allow us either to knock down pre-existing neutralizing antibodies so you don't get screened out in the long run, or if we needed to down the road, so, hopefully some many years later, we could redose these patients.
Yeah, thank you. I mean, it's always on our minds, the what if, right? But as we're talking about immunity, Doug, I think one of the questions that all of us has for children who screen for a study and are found to have antibodies, and obviously you have your parameters and your methodologies to screen for that. But when a family gets this news, it feels like the diagnosis all over again, and it is so devastating, because I think all of us in this community know that, or believe that, restoring dystrophin, whether it's a microdystrophin or through an antisense or some way somehow getting dystrophin in the muscle, is the holy grail, if you will, for lack of a better term today. So I guess as you gain more knowledge through these studies, do you feel that there would be a time when you would be a little more flexible with those parameters and maybe be able to loosen them? Or do you feel that the parameters that you've set are going to be set in stone forever based on your knowledge?
A couple of thoughts on it. First, I want to frame it so people are aware of the risk, but also frame the risk. So we're not talking, I know we've seen literature on some other AAV serotypes that have what appear to be very large screen out rates. For RA75 so far, we're looking at something that would be less than 20%, like in the mid-teens. So it's still a very significant issue, of course, and it doesn't diminish the impact that it has on the individual patient and their family if they do screen out. But I do want to point out that it's far more likely than not that a patient that hasn't been yet tested isn't going to screen out. So just for planning purposes, I think it's important to understand that. I don't think the parameters are going to change, because I don't think we're being exceptionally conservative. I think we're setting the parameters in the right way. Remember that slide on the things we're looking at starts with the proposition that safety comes first. And I think if we change the parameters in ways that we didn't think were supported by the science, we might be in a position where we're putting patients at risk without a benefit to them. What I do think is hopeful, and again, I just want to go back to that forward-looking statement slide yet again and continue to remind us that, we are guided by the science. And, I've got a silly T-shirt that I wear sometimes. It says, "Science doesn't care what you think." And so, my views on science and predictions on science, may or may not come to fruition. But I would say, I think in the scientific community generally, there is a strong belief, probably bordering on confidence, that at least with respect to pre-existing neutralizing antibodies, we are going to find solutions that bring this therapy back to dosing for kids that would otherwise screen out of neutralizing antibodies. We have a number of approaches that we're working on inside of the organization. We entered into we've got our own organic approaches, and we've got our own approaches with some really smart scientists over at a company called Honda, another company called Selecta, with the goal of being in a position to be able to either redose or knock down pre-existing neutralizing antibodies. So I would say to those people who are sitting here today and, wondering if, goodness, wouldn't it be a horrible irony that it turns out this works? It turns out we turn the cards over next year and the results look fantastic, but then my child gets screened and they have a pre-existing neutralizing antibody for some reason, and I am robbed of an opportunity. I understand how potentially devastating that could be, but I would simply remind everyone that the statistical probability isn't high that that's going to happen to you, and in any event, that doesn't mean that it's over. In fact, we're working like mad to find a solution that would allow us to figure out it would be some kind of co-dosing or some other approach that would allow us to dose children who even screen for pre-existing neutralizing antibodies. So I think even in that situation, there would be hope.
Right. Thank you. But it is just hard. I don't disagree with you that it's probably 15 to 20%, although, we still have to understand that. But it is, if you're in that 15 to 20%, it's pretty devastating. So can we go back to slide number 12, please?
While you're going back, I just want to make sure you know, we agree with you completely, and this is the top, this is one of our top priorities. Certainly getting this therapy to a place where we've proven that it works and regulators will allow us to bring this therapy to children in the U.S. and around the world is a top priority. But the ability to redose is not redose, but knock down neutralizing antibodies is enormously important to us. We have a mantra in this organization. We don't want to leave anyone behind.
Yeah, no, I can understand that. Nor do we want any young man or for those women as well with manifesting weakness. We don't want anyone to be left behind. And as you have said, quite appropriately, time is everything for these patients. So now we look at your clinical development program, which is really terrific and certainly robust and never going to be soon enough for any of us here. But in the study 101, you used, I believe, 2e to the 14th was your dose. Is that dose going to be consistent across these studies?
Yes, it is. We're very, not only are we pleased with the results that we've seen thus far, but there was a lot of work done from a preclinical perspective, looking at animal models and the like, to come to the right dose. And there's a lot to think about here. We were able to get to a dose that in preclinical models showed us that we had a great safety window. We could dose this up in animal models 3 times higher, and still not have a significant safety-related issue, which of course is paramount issue. But also, when we get to about this level of dose, the animal models predicted that if you dosed even higher, you're just putting more therapy into the children. You're not getting a benefit back out of it. And the good news, at least in the first trial that we've seen, is that it certainly bore out similarly. And we've got I'm going to get super complicated, but there's another gene therapy we have for another devastating neuromuscular disease called limb-girdle 2E. It is in many regards shares a lot in common with Duchenne. We've dosed 3 children in that study with the same viral capsid and the same promoter that we're using here, but a different gene. So from a safety perspective and an expression perspective, a lot of similarities. And good news there, they got expression levels in that therapy that were approaching normal, and they had very, very well tolerated, and it appeared to be very safe. So we're growing in our confidence about the dosing that we're doing for the children.
Okay, that's really good to hear. And then if we look at study 102, 41 patients, it was a crossover design of 1-to-1, 1 blind to 1 person inactive, right? And I think that study is completed. Is the data locked, or is that study still open and ready for the data to be locked?
No, it's still open. It's still blind. We still have a couple of functional visits remaining. All of the kids for the main analysis have been dosed, and a significant portion of the kids on the crossover have even been dosed as well. But we still have a few functional visits. So that study will complete before the end of this year. It hasn't completed yet. And then it will be locked and, of course, quality controlled. And then we will release it in the first quarter of next year.
Okay. And was it the same protocol in terms of you biopsy these children at 3 months, baseline and then 3 months, same as Study 101?
Yeah, yes, the same as Study 101. The one difference in the protocols that we made, in addition to taking out some of the limitations, is that we extended the steroid protocol from 30 days in trial 101 to 60 days in trial 102, and we'll see. The goal here was maybe we could even avoid these transient liver enzymes if possible. It turns out we did the same thing with this limb-girdle type 2E, and at least in those 3 patients, very small set, it works. So maybe it actually works in Study 102 as well. But other than that, very similar.
Yeah, and one thing to add, Pat, is in that study in 102, the boys also get a third biopsy at week 60, so roughly a year and a bit after in their rollover and their crossover. So these boys actually were great. They volunteered to get 3 biopsies.
Wow. That is terrific. As you well know from meeting with various patient groups and certainly the Community Advisory Board in Europe, recommendation is 2. So those are very brave parents, and we're really thankful and for their willingness and their children's willingness to participate in that. So now as we move into the Endeavor trial 103, this is expected to start before the end of the year. So that's terrific. And you have said, Doug, a limit of 10 patients. You're going to start with the littles, the 4 to 7-year-olds, and then you're going to add some non-ambulatory patients with no age restriction. So my first question is there a weight restriction?
There is no weight restriction.
Okay, and you're not at all, and no age and no weight restriction on that.
Nope.
So,
the first cohort is 4 to 7, but the non-ambulatory cohort wouldn't have an age restriction. And trial 303, this sort of large placebo-controlled non-ambulatory study, will not have an age limitation.
Okay. Well, that's pretty terrific news. So you can go, we often see studies that end at age 18. And as you know, we have many young adults over the age of 18 that certainly are very anxious and interested in participating in studies and certainly receiving therapies. So as we think about Study 103, you're going to do 10 patients only in that. Is this based on what you see with the flexibility to expand it upward to maybe 20 patients or 15 patients as you see fit, just to explore that? This is with commercial manufacturers. So as you learn about this commercial and prove that it is acting exactly the same as the nationwide vector facilities material?
I think a couple things. The 10 patients was chosen. Look, for us, the goal is a combination of getting scientifically rigorous data and getting it in a time frame that's relevant to the patients that are waiting for it. And we took a very careful look, and 10 patients will give us the data we need to explore the expression and safety of our commercial material as a validating study. We will add patients to it or do a separate one like it for non-ambulatory patients, and we'd like to start that as soon as possible. We have conversations we need to have with the agency on that. The real goal for us is to also get going on Study 301 and 303. So really after Study 103 and we get initial read on Study 103, we want to move as fast as possible in Study 301 and then Study 303, which are these large multi-center, multi-country trials, placebo-controlled for 4 to 7-year-olds and also for non-ambulatory patients.
Yeah, for sure. Those 301 and 303 will be blinded trials, right, with a 1-to-1 blind, or are you doing a different blind?
Exactly, blinded and 1-to-1 placebo to active.
Yeah. Okay. So back to 103. One more question on 103. How many sites are going to be involved in this 10-patient study? Is it one or just the 2 sites you're using now?
It's going to be, it's a multi-site trial. It's U.S. specific, but multi-site trial. So we have, I think, probably as many as 8 potential sites in the trial. whether we'll use them all or not, given that it's only 10 patients, we don't know, but we want to have maximum flexibility to move as fast as possible.
Yeah, and certainly to open these sites, right? Because I know that in other studies they've had, I think what our data shows that between 2 and 22 months to open a site, and I can't imagine what the pandemic has done to the academic center. So you'll want to do whatever you can to get these sites up and ready to go so that they can then embark on the 301 study. So the 301 study, embark, how many sites are you thinking? And this is global, is that correct?
This is global. This will be U.S., Europe, Asia, and I think we're considering Latin America as well. We don't have all of the sites. We've done an enormous amount of work, our clinical operations team, but we don't have all the sites, sort of a list of all the sites, because we first got to get through that process with them and get, not only get it checked off from us, but get them to approve it as well. But this is probably as many as, now, Teji, you correct me when I get this wrong. This is probably as many as about 30 sites or so, I think.
Yeah, so we're upwards of 40 now to allow for that slightly larger sample size and also to mitigate against any restrictions from COVID. So just above 40.
Yeah, so here's a struggle, right? 301 is 75 patients, the little kids, 4 to 7-year-olds, which is terrific. And then the Envision site is 80 patients, 1-to-1 blind, and these are non-ambulatory patients. I think this community is really worried about those who fall in between. When we look at boys at the age of 8 years old who, the natural history is between 8 and 13, and obviously there's a mean in there, but that's when we see these young boys are fragile. They start slowing down, they start falling, they really start to become that in the transition to losing ambulation. So I think that this is a population that needs to be considered because they are fragile, because maybe with luck and whatever else it takes, prayers, luck, and real possibility, wouldn't it be pretty amazing with the expression you've demonstrated to treat this population so that they perhaps could have much more time on their feet? And so I find that there's this gap in between these two studies. So I wonder if you could address that.
Yeah, it's a really good question. Look, it's a struggle for us. The issue is, of course, let me be clear about what our goal is. Our goal is, these trials are a means to an unbelievably important end if this therapy works, right? And the goal, of course, is to find a way to generate the evidence for this therapy that gives us a broad label so we can bring this to the community as fast as possible. That is the goal behind all of this. And one of the risks that we have is if we study every potential age, we may just delay the opportunity to bring the therapy to the broader community. So as an example, people have often looked at this and said, why 4 to 7? That seems fairly narrow. It's narrow because if you want to take a small data set, if you want to see a statistically significant difference in a short period of time, you've got to narrow the window so that when the therapy is working, you can see it and the regulator can see it as well. We have we haven't put them on the slide. Here's some other thoughts for trials that we'll be thinking about over the course of next year. But our primary goal, the reason we're being restricted in these early studies, is that we want to bookend what we've studied and then say to the regulators, it's time for us to get an approval to bring this to the community, and we want a broad approval. And what we've shown you is, we've shown you that it works, and mechanistically it will work for all patients. That's certainly that would be a logical assumption, and it's been the assumption that at least CEDAR over at FDA has made with respect to dystrophin inducing therapies like this. We've shown that it's safe in older kids. We've shown that it's safe in non-ambulatory and larger kids. And so, we should get a broad label. Let's go as fast as possible. And that's kind of been the tack that we've been taking.
Yeah, I'm not going to argue with that. It's just that little fragile population, that fragile population is fragile, and years make a difference, right, as your regulatory process takes place. So I think, if it were up to us, we'd ask an open label of those patients to see what we can do. So just obviously there's lots of issues around there and concerns and work that would have to be done. But I think we can't forget that population that is fragile and that could significantly benefit, as all populations could significantly benefit if microdystrophin is useful. So that brings me to the regulatory process. So if you envision your regulatory process forward, you're thinking about, I'm making an assumption here, you're thinking about taking the data from the trial 102 of 41 patients with a 1-to-1 blind. If it looks robust, you're going to use that with and combine it with the safety of trial 103 and say this material is acting the same as the previous material produced by Nationwide Children's Hospital, and ask the FDA for the pathway forward, or, because obviously in your products that are approved, you use the accelerated approval pathway. There was worry on all of our parts that payers wouldn't pay, but it turns out that with, in some cases, pushing and prodding, those payers are paying. So can you just sort of tell us a little bit about your thoughts around the regulatory process?
Sure. And you'll notice that I haven't said this is the particular pathway. And the reason I haven't said that is because I want to be assiduously accurate and say that we don't have an understanding with the agency of exactly the pathway. What I can tell you, and there's a good reason for that, which is we want to have that dialogue with the agency with the evidence in hand. It's one thing to have a discussion with a very busy, overburdened agency about theoretical issues. It's a very different world in which you have data in hand and strong evidence. And let us assume that we're successful. And that, of course, is a big assumption. We'll find out when these trials read out. But if we were successful, we would have, by the middle of next year, between study 102 and study 103, we would have evidence that our therapy was efficacious, that it was helping these kids and moderating this disease and hopefully transformative. We would have evidence that the therapy is safe in these children, and we would have evidence in how the commercial material, the material that we would propose to bring to the community with the new manufacturing process, how it performs both from a safety perspective and from an expression perspective. And then we'll sit down and we'll talk to the division, first the FDA, and of course then agencies around the world with our partner Roche about the pathway forward. You can imagine that our goal will be to bring this therapy to these kids as fast as the science permits us to do. I would also note that this will be different than, for instance, Exondys at the time that we got approved. Exondys was approved on a biomarker, which was the expression of a truncated form of dystrophin as a biomarker. We will have by next year, out of a well-controlled trial that is blinded and placebo-controlled, results on the function of this actual therapy and the efficaciousness of this therapy. So, I think by the middle of next year, I can't make promises about what the pathway will be, but I think there's a real chance, if we're successful, that we could have a robust set of evidence that this therapy is a meaningful benefit to children with Duchenne muscular dystrophy. And you can imagine that we're going to be pushing to move as fast as possible to bring that therapy to the patients that are waiting. Because we all know, and I hate to keep saying it because it probably eyes roll at the obviousness of this statement, but time is not on the sides of the children that we serve, and we feel an enormous sense of obligation to find a fast pathway to do something with this therapy that works.
Yeah, no, I think one of the big questions of FDA is what is the microdystrophin and what does it mean in terms of functional outcomes? So I think being able to sort of unwrap that a little bit and explain it, or obviously scientifically explain it to them, would go a long way in sort of proving, if you will, or providing them the opportunity for a path forward. In the past, your other approvals have been through Center for Drugs, CDER, right? And I think Billy Dunn and Dr. Basings have all gained significant comfort with Duchenne and its progression. And I think the CBER folks, the Center for Biologics, will come along as you and others are able to talk about microdystrophin and its overall effects. I'd love for you, switching gears just a little bit. What about a sibling protocol? What about if I have a child who fits your protocol and a child does not? What kind of conversation are you going to have with me about if my child is included in your study, as he sheds the virus, what risk is the other one carrying? And second, if my other child is older, younger, or even fits the protocol? How will we separate them? How will we treat them? And how will you treat if the child doesn't fit into the existing protocols? So that's a lot, but I think you can manage it.
So, Teji, why don't you tell us where we are right now in thinking around siblings? Because it's obviously a very important issue for families.
Sure. No, great question. I know, Pat, you've raised this before. So a couple quick things that, obviously siblings are allowed in the study, but they're not at the moment assigned to the same group. So we're still looking at that or working to see if that's feasible. In terms of the shedding information, in terms of the non-clinical studies, we actually haven't seen that shedding leads to antibody formation in a non-exposed animal, so in a non-human primate study. We are kicking off with the 103 and the 301 a seroconversion study where we're trying to get samples from caregivers and other close family contacts to understand if there's actually, a shedding leading to antibody conversion, because that'll help guide some of our decision making. In the absence of that data, using data from other AAV trials, we do have precautions for the first couple of weeks to the first month. There's specifically a straightforward hand washing, contacts, limitation in terms of actually exchange of bodily fluids, that sort of thing, but there's nothing really, really extreme. But to your question, if one child's treated and the other's not treated, is there a risk? We think it's negligible. We're trying to put some numbers around it. We don't have a definitive protocol for how to give access to the non-treated child yet, but that's still something that we're trying to explore and consider. We'll have more data when we finish the 102 about, actually if there's a positive benefit. And I think we'll bring data from 103. One of the things that we're actually doing in 103 before we start the phase 301 or 303 studies, we're actually getting our shedding data out of 103. So 103 will inform on that shedding duration, and then I think we'll address in 301 or 303 the actual precautions that we need to do that's evidence-based.
Yeah, well, thank you for that. Just long ago, I argued with Jerry Mendel when the steroid studies began that if you have siblings and they fit the protocol, it should be N equal 1 in terms of the arm, because you can imagine if one child is doing better and the other child is losing strength, just the impact on that young man to watch his brother, and then the impact on the family. So I'd vote for that, and I'd also vote for, since I'm not making Sarepta's rules, I think that you should keep the 103 open, and if a sibling or if the family with more than one child comes and one fits the protocol, you could ship the other younger or older into the open label. But again, Sarepta, I'm not making rules, I'm just making suggestions, because I think as a family, when you have two boys, it's really hard, and you really want them to have the best opportunities and the most for them both, not just one. So I know that you all have a hard stop in 15 minutes, so I'm going to go to some of these other questions so that we can cover most. I'm sure there will be many more, and hopefully once you get to past the first quarter, we'll all be asking you to talk about the 102 study and what you've learned. But so, as you know, you have any number of young men that are and some women that are on your antisense oligonucleotide therapies. Do you see a day when they would be? Do you see any issues with putting a microdystrophin next to that truncated dystrophin expressed with your antisense? And how do you see us getting to combination therapies?
That's a great question. A couple of thoughts on that. One, the interesting aspect of that is that there is already literature that Sarepta wasn't involved in, but there was literature from Dr. Voigt, as you know, over in Europe, looking at the pre-treatments in advance of gene therapy using a PPMO, interestingly enough, a version of what would be our next generation version of PMO. And then looking at gene therapy and finding, at least in the animal models that Dr. Voigt looked at, an actual benefit to having both of those therapies and to pre-treating with the PPMO. So let me say without overpromising, one, I imagine a day we got to do the research on it. We got to see if we're right. I think there's a potential for a combinative benefit between one of our RNA therapies, either PMO or if it works PPMO, and gene therapy. We have work to do. In the shorter term, and I'm glad I can't see Teji right now because I'm sure he's rolling his eyes fearful about what I'm saying. I do think that one of the things you won't see on the slide, but we've got to consider as an organization in the not too distant future is looking at studies that take patients who are on a stable, maybe even long-term version of one of our current therapies, and then seeing what gene therapy looks like for those patients, so that we can make some long-term decisions. Like should patients who have been on a long-term version of a Sondes or a Teplerson, or if it's approved Amondys, should they be washing out, which they have to currently do as part of the protocol? Should they in the long run be washing out in advance of gene therapy, or would they be better off not washing out? Would that be the best answer for these patients? But that's going to require some research and some study, and it is something that the team is exploring right now. It's not on the slide because it's kind of early in the exploration of that, and it's been a challenge to the team to consider how we might do that.
Yeah, thank you for that. I was raised in a German family where they always thought more is better. So I'm hopeful that it is true that those microdiscs, along with an expressed truncated dystrophin, would be complementary and synergistic. So that would be terrific. As we move forward, this pandemic has been such a struggle in so many ways for all of our families, isolating, worried, concerned about what happens. But now, I'm not saying we're emerging from it, not at all. I wish there was a magic wand. But as we move toward a vaccine, how can we think about vaccines, right? Is a child in a study able to get a vaccine? Is a child in a study to be vaccinated prior to joining? And if we look at this window of 4 to 7, and he's 7 3/4, and he needs to be vaccinated in a 50-day window and loses the opportunity, I mean, I can imagine the devastation around this. So I'm sure you've thought about vaccines, what the risk is, what the benefit is, how to manage them, and even there's been questions around, I think it's the Oxford Moderna study that's using a virus for delivery. So if you could comment on sort of the vaccine strategy, the worries, I don't think there's any concern with the virus that Oxford's using as it relates to the RH74, but if you'll comment on that and talk to us about how you're thinking about vaccinating or not, or the timing, so that we don't have parents reluctant to vaccinate their children because they're depending on at least being screened and considered for gene therapy.
Yeah, I agree. I'm gonna turn to Teji to answer sort of what we know in the early days of this. Before we does answer it, I will just echo what you already said. Let's clear, apart from some of the just general risks for the trial and what that might mean and vaccination, we should know that the fact that the Pfizer study, the Pfizer construct and the Moderna construct are mRNA constructs, that in and of itself doesn't have an impact on this issue. There's nothing about getting that therapy that, for instance, would generate a pre-existing neutralizing antibody to our therapy that would rob a patient of the ability to get our therapy. But Teji, you probably have some thoughts on at least the early days thinking about what we would do with a vaccine as the vaccine comes out in some of our trials.
Sure. Thanks, Doug, and a great question, Pat. So just a couple of quick things. For currently approved vaccines that are not COVID-related, our trial does allow for vaccinations, and we have some restrictions on when they can be given, not because of the safety concern of the vaccine, but because that steroid regimen that we're boasting up for the coverage during the infusion may actually make the vaccine less effective. So for our trials, there's a limit on the live, how much before, and non-live. So 4 weeks for live, 2 weeks for non-live, prior to the infusion, and then that 30-day, that 60 plus 30 days or 90-day total window where we don't recommend vaccination because of our steroids. So it's not the safety, it's to make sure that the vaccine that's given is efficacious. So we expect the boys to have their routine vaccinations. With regards to the COVID, great question. One little disclaimer is that, of course, I don't really know a lot of details about that because it's yet to be approved. And once it's approved, they'll come with the data on what's exactly happening. It'll come with the CDC recommendation of the schedule for it. But as Doug pointed, for the mRNAs, they're going to produce a protein, that's sort of like the spike protein that should not have any problems for us. We don't expect that we'll have any sort of cross-reactivity to it. For the other ones that are coming up, the adenovirus ones, and then potentially the MGH AAV ones, it would depend on the stereotype. So again, when we have more of a detail on exactly what it is, that will determine whether or not there's a cross-reactive antibody. But again, we don't suspect that. I mean, the Johnson & Johnson one is in AG23. But again, when that data comes through, it'll give us an idea of there is a antibody that's cross-reacted beyond that. Typically, the other vaccines are also proteins that have some reactivation, and some are already on the basis of a virus, and we haven't seen any issues with that. So I'm optimistic that we're going to have a way to define and how to do this, and then if there is a delay, it's either pre-dosing or there's a delay, get the infusion, and then we get the vaccination after the steroid period is finished to allow for the full effectiveness of the vaccine.
Yeah, thank you, Teji. I was worried about the impact of steroids as well, and I just got a notice that Governor DeWine, who's Ohio's governor, said that he will have the vaccine, the Pfizer vaccine, in Ohio by December 15th. Now, I don't know if that's wishful thinking, but he made the announcement just today. So, yay us in Ohio, and hopefully it'll be many others, and hopefully it's effective. I know you both have a hard stop. So, first of all, thank you for this. Just one comment. You're still working in the area of CRISPR. How do you see that coming forward? Do you have anything to sort of give us a little tidbit about what we can learn about that next and where you are?
I think CRISPR-Cas9 is a really exciting technology, and I think if anyone wonders how exciting it is, we should remember that I think within the last month or so, two women who are luminaries in this area received Nobel Prizes because of the CRISPR-Cas9 technology. Just to remind everybody what's the difference in the broadest of strokes between what we're talking about here in CRISPR-Cas9. You'll remember the beginning of my discussion, I said that we don't fiddle with the gene itself, the Duchenne gene. We go over and we just ignore it and we put in a new gene that makes the protein that we want. CRISPR-Cas9 does something very different. It delivers essentially a combination of a GPS unit and scissors into the cell where it will edit the actual gene and create, hopefully if all works well, allow it to start making a protein again. Couple things. One, we're very committed to this as a potential technology in the future. We actually have a gene editing, which is based on CRISPR-Cas9, a gene editing innovation center that we built in Durham, North Carolina, under the leadership of Dr. Charlie Gersbach, who is brilliant. I think there's a lot we have to think about with respect to CRISPR-Cas9. It is still in the research phase for Duchenne muscular dystrophy in particular. One of the issues with the CRISPR-Cas9 is you've got to find a way to both get it to be a proficient in editing genes, but you don't want it to be so proficient that you have off-target effects. That would be devastating. So we've got to be very careful about that. I think one person's view, at least, and I could be very wrong about this, but I think using AAV to deliver full body infusions for gene editing may be difficult because you get this one opportunity to do this. And it's not like gene therapy where you can provide this massive infusion and then get great gene expression everywhere. You've got to make sure that you get great gene expression, but you don't get off-target effects. So finding something else, some other approach, and we're looking at them, exosomes or lipid nanoparticles, where you could dose over time and edit over time would be an interesting area, but we're looking at that. It's still a research project. We're still years out from that, but we're considering it. One other thing you need to remember is if you're going to edit the genome with respect to Duchenne muscular dystrophy, as we all know, there are an enormous number of different ways in which the gene can mutate. And so we would be talking with gene editing that we would be going back to a more bespoke approach where we would be editing essentially area by area to be putting subpopulations of children with Duchenne muscular dystrophy back in frame. Now, Dr. Gersbach's got a really fascinating approach where he does an edit of a larger region of the gene, which would allow about half of all children with Duchenne muscular dystrophy to be put back in frame. But it is, I would say, it is an exciting technology, but we are still some ways off from translating that into an effective therapy for children with Duchenne muscular dystrophy. At least that's what I think today.
Thank you. I too, looking at the research, know that that's very early, and hopefully as we learn more, that will also evolve and become more efficient. So I know it's 2:30 and you'll have a hard stop. I can't thank you, Doug and Teji, and all of Sarepta enough for this webinar today and this discussion. I hope it's been helpful for the community. For all of you online, it's nearly Thanksgiving in the U.S. And I think all of us in the U.S. are grateful for this community and all of the companies and all of the people working in it. We want to end Duchenne, and we want to do it as soon as possible. So thank you again. Happy Thanksgiving to you, and we will see you very soon. With that, we'll stop the webinar. Thank you again for joining us. We'll see you soon.